Well, good day, everyone. I'm here to present to you a tool that we have been developing in the last year. It's called Target Pathogen 2. It's an automated bioinformatics tool for drug discovery in bacterial genomes. First, I want to talk about where we are right now. In terms of the number of bacterial species, it has been growing in the last year. The number of bacteria that we have classified has grown almost 20 times. If you go to 2005, we just got 30,000 different bacterial species, but if you go right now to the NCBI, you will find almost 600,000. On top of the new species that are being classified, the ones that we already know are causing us trouble because just in 2019, the number of people that died from drug-resistant problem by the World Health Organization was almost 5 million people. And also, we got the problem of the antibiotics. The pharma is not developing antibiotics quick enough because it's not cheap, and they don't get the returns that they want. So we hit a peak in 1984, and we hit bottom in 2009. Now we are recovering, but we need to develop some tool that allow us to accelerate this process. That's why our goal is to develop a sophisticated bioinformatic analysis tool that enables a simplified user experience for non-bioinformatic experts. So any expert that works on a particular bacteria can access the bioinformatic tools that we all know, but we know that it's a little difficult to get started. Also, the aim of the platform is to identify potential drug targets efficiently and establish a robust and scalable database. For that, I want to talk to you about the pipeline that we have developed. The only thing that we need to run the pipeline is the genome of the bacteria in the GBK format. And from there, we got the automated annotation pipeline. This pipeline goes to different databases from Go to EC to PFARM, Interpro, all the different databases that allow us to get information about the genome, and also runs different tools or different protocols like BLAST, for example, for get if some protein is essential for the bacteria, or if the target that we are looking for is present or has an off-target in the human proteome or in the gut microbiome. Also, we got a module for reconstructing the metabolic pathway of the bacteria. So all of this, I said that this is called Target Pathogen 2. That's because Target Pathogen version 1 has been working for several years, and what we are doing right now is to actualize it to the new technologies. But from here, almost all of this was present in the first version in some manner, but the things that we are adding to this version, for example, is the use of novel tools that are being developed right now. I think PSORT is not so new, but the platform is able to absorb the different tools that are being released every day. For example, PSORT is a tool for calculating the cellular localization of a protein. So one of the things that we added is the use of the AlphaFold database to build the structurome of the bacteria. And from those structures, we run some tools for packet recognition. One of them is FPocket, and the other one is P2Rank. They do the same thing, but in a different way. For example, here we are seeing a portion of a leucine ligase from Neisseria gonorrhea. We can see that P2Rank is able to predict the surface of a pocket that's able to bind to some drug, and F-pocket is able to calculate the volume of that pocket. So from here, we got the pocket prediction information. And another thing that we have built is the search for the known ligands of the bacteria. We are doing that searching in the Campbell and in the PDB bind database. I will be switching between the pipeline and the website that we have been developed. Here's how it's look, the known ligands from the bacteria. Here you got a representation, the name, and you got some links to do the research about those binders. So one thing that we are doing right now is to corroborate that the pocket prediction is actually predicting places where some known ligands are able to bind. So from what we got so far is the pocket prediction and the known ligand modules. This is the results that we got so far. With the first version, we were able to run this pipeline for more than 30 different species of bacteria, including tuberculosis, Pseudomonas, and Klebsiella. And we published more than 10 scientific papers using the first part of this pipeline. We are currently working on the paper for this version. Here we got a picture of one of the sections of the website. Here you got the locus tag, that's the primary key for the database, the description, the gene name if it's available, and in the last one we got the druggability calculated by the pocket prediction tools. The website, this part of the website has two main features. The first one is it enables you to filter out the properties that you don't want. For example, here we're only seeing the proteins that got a medium or high score in the pocket prediction, in the druggability by the pocket prediction tools. And the other feature of the web page is that it enables you to select the different properties that you are looking for. So if you are an expert on some bacteria and you know that the delivery system for the drug that you are researching is not very good, and you really need that the target is located in the cytoplasmatic membrane, well, you can assign a very high coefficient in the score function that you can construct. So every expert has the ability to really pinpoint what they are looking for in the research. The technology that we used to develop the website was a PostgreSQL database, a relational database, to ensure that all the database is homogeneous. And to develop the web site, we use Django on top of some other libraries that I don't need to mention right now. One of the things that really helped us with the pipeline was the use of Docker to containerize all the different modules. So you can use the different modules if you want just that. For example, if you just want to run the structural module or the binders module of the pathway recognition module, you can do it easily and on any computer. And also we use parts to parallelize all the calculation and to enable us to easily scale the project when we got some more infrastructure. That's the work of Parts. I really recommend you to check on that if you're doing automated pipelines. So this is what we got so far. The pipeline enabled us to calculate all the genome, the proteome, the metabolome, and the structurome in an automatic fashion. So what we want to do in the future, in the next semester with this pipeline, is to populate the database with all the information for the pocket prediction and the known ligands to build a list of features that enable us to develop an AI model capable of discover novel ligands. We're aiming to a point where any researcher got, for example, some drug that it's approved by the FDA because it's used in another pathology, test if that drug is able to bind to some of the calculated pockets of the bacteria. So, in summary, Target Pathogen enables you to do sophisticated bioinformatic analysis without requiring any technical expertise for the user. It ensures the reproducibility of analysis across experiments, and it streamlines laboratory processes by filtering out less promising targets, saving valuable time that we need, and establish a robust database to train the AI models for novel drug discovery. I want to thank to this university, the Universidad de Buenos Aires, to GabanaNet that enabled us to get to this point, and to all of you for listening to this. Thank you.
Any questions to Gabriel? Could you come here, please, to talk to the microphone? Sorry for that, but we have some people watching online and they will not listen if you ask from there.
Gabriel, thanks for a nice presentation. My question is, you mentioned that you are planning to implement AI-based algorithms for recommendation of new ligands. Do you think that they also can be helpful in the process of selection of target, the implementation of an AI model to use the features that you calculated before to recommend the better targets?
Yeah, that's a very good question. Yeah, we may do that in the future, but right now we're really focusing on finishing this module. And if we got good results, maybe we do what you're doing, what you say. Yeah.